We're here in Wytham Woods, which is actually a massive area. It's about 1,000 acres, which is a lot larger than people often think. And it's not just woods, and we do have some fantastic ancient woodland, but we also have a huge diversity of other habitats, such as calcareous grassland and even a fen down in the south east of the site. Wytham, in a sense, is an unparalleled site in research. It's been under the possession of the University of Oxford for almost 100 years now. And alongside that, there's some very long-term ecological experiments. So there's a whole raft of different research going on in Wytham Woods, including work on birds and badgers and plant surveys, and now the Tree of Life projects looking at the genomes of all these different species. Here at Wytham, as part of the Darwin Tree of Life project, we have more of a focus on arthropods, so insects, spiders, millipedes, that kind of thing. So I'm an entomologist, so I just love going out finding and identifying stuff. So I found out about a position as a research assistant on the Tree of Life project, which sounded like the dream job, and sure enough, it's been a fantastic few years going out finding all new species that I'd not seen before in the woods, and hopefully many more to find. We found so many cool species that we didn't expect to find, but we're hoping to get many more as we continue to go into new habitats and find new target groups. So we've had some fantastic moths, such as the Clifton nonpareil, which is also known as the blue underwing. It's got these fantastic bright blue hind wings which flash out as it's flying. We've also found some really interesting beetles associated with deadwood, some saproxylic species. And my particular favourite highlights are the bees. And we've had a couple of really interesting species, such as Nomada hirtipes, which is a nomad bee, which is a cuckoo parasitic bee on another species, Andrena bucephala, which we found this morning. We've had a really great morning collecting samples for the Darwin Tree of Life project here in Wytham Woods. We started off the day by looking in some calcareous grassland, so we're in an old quarry and on other patches of grassland, and we did some sweeping, so just sweeping our net back and forth over the vegetation to collect all sorts of different species. We did find one of the Wytham specialities, a really rare beetle. It's in the family Apionidae, so seed weevils, and it's a tiny little thing called Pseudoprotapion astrolaugi, and you only find it feeding on wild licorice. And I've been desperately trying to get a photo of a live one on the host plant, so I managed to get that photo this morning, so I was really pleased. We've also seen lots of other species of spiders and flies and beetles. We've also done a little bit of beating. So we were beating some young hawthorn saplings, and we had all sorts of interesting species that dropped out into our net. So we had 10-spot ladybird, orange ladybird, some interesting ant-mimicking true bug in the family Myrridae, and an interesting beetle as well, which hopefully will be a new species for genome sequencing. We're hoping to get many more as we continue to go into new habitats and find new target groups. And these largely reflect groups that we haven't been able to drill into up to this point. So this is things that tend to be quite small or quite difficult to identify. So we're bringing in people with that specific expertise to look at things like springtails and soil mesofauna and myofauna. Just look at what's flowering. So, for example, the bluebells here, or we'll also be looking at flowering trees, which can be fantastic for pollinators. And just standing and watching, seeing the species coming in, land on the flowers, and then we can then catch those with an aerial insect net and identify them. We'll have a little look along some of the more open rides where a bit more sunlight gets in, so you tend to have greater floral displays there. And also a lot of these pollinators like to bask. So anywhere where there's sunny dappled spots, they'll sit on a leaf warming up in the sun. Weather and time of year is really important when collecting insects, but more for some groups. So some things like beetles, saproxylic beetles, we can collect all through the winter, and things that live in fungi as well. Whereas other things like pollinators are a really good example of that. Butterflies, another one. They are really dependent on the temperature, so we're only be able to find them on sunny days and even sunny times during the day. So if a cloud comes over, they'll all disappear, and then within minutes of that cloud going, suddenly they'll all be out and active again. So we're out in the field, collect a sample, we then take that back to our lab facilities, which are in the woods itself. We can then flash freeze, and we're flash freezing everything on dry ice. And then once we've got those samples, we then ship those. First of all, we send legs for DNA barcoding to the Natural History Museum, and then when the identification has been confirmed and we can track that specimen, those samples go off to the Sanger Institute, where they undergo DNA extraction. And then once we have the DNA, that can go through DNA sequencing. And then once we have the DNA sequences, that can go to the assembly team, which then build together these fragments into the complete genome, which is then published. And all of the steps along that process are completely open access and available to the public to log on and look at that data. So one of the really nice things that we're doing here in Oxford is, as well as going out and finding all these genomes in Wytham Woods, we're also beginning to analyse the genomes at the other end of the pipeline. So once the genomes are produced, we can then start to ask really interesting biological questions about what those genomes look like, how they have evolved, and how that might relate to the organism that they've come from in the context of the woods. There's lots of stuff that we can learn from genomes. We always talk about it here at Oxford as a two different ways of doing it. The first of which is looking at the genomes and looking for interesting patterns in those genomes, then trying to work out, well, what effect on the biology does that have? The other is to take the opposite approach and find an interesting bit of biology and then look at groups of organisms which have that, and then trying to go into the genome to see if there's any differences between them. So for example, we're looking at the opsin genes which regulate eye development, and we're looking at between day flying and nocturnal moths to see if there's any differences there. And then an example of the other approach would be looking at the homeobox genes, which are important developmental regulatory genes. And then we've found really interesting patterns within moths, where some moths have these crazy duplications with these massive genes, and we don't really know what the function of that is, or if it has a function. It's also very exciting for conservation. Because these are providing reference genomes, that is the essential first step for a lot of resequencing projects. So if we want to work out how related a population of endangered species are to each other, or look at genetic bottlenecks, we need to have the reference genome to find the loci within the genome that we can then go back and sample. And once you have that reference genome, you suddenly unlock this whole other branch of methods very cheaply and very quickly to resequence specific parts of the genome. There's lots of kind of more applied discoveries that we can make from these genomes. In a sense, we don't know what we don't know, so there could be all sorts of potential benefits for new medicines or for new technologies.